Welcome to Zoology class. We have been discussing different processes or different steps in the central dogma. And in the last class, we have discussed about transcription. That means the process of transferring informations from the DNA strand, from a small segment of the DNA strand to an mRNA, or the synthesis of mRNA which carries the coded informations of the DNA strand regarding the amino acid sequence of a protein. Okay, now the next step in the central dogma is translation. 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 So translation is the process of synthesizing a protein. Not simply any protein, but a protein whose amino acid sequence is determined by the DNA and transferred to the messenger RNA in the form of genetic codes, triplet codes. So based on the triplet code on the messenger RNA, now a protein is to be synthesized in the cytoplasm, and that protein would act as an enzyme to carry out a biochemical reaction which results in the expression of a phenotype or phenotypic expression. Okay. Now, what are the components for this translation? What are the substances or what are the molecules or organelles required for this transcription? There are certain components required for translation. What are the components required for translation? Let us see what are the components required for translation. The first one is mRNA, of course. There should be an mRNA which carries the triplet codes or genetic codes for the amino acid sequence in the protein. That about mRNA we had already studied in the previous classes. And an mRNA, suppose this is an mRNA, and this mRNA has triplet codons, and it has untranslated regions or UTR, untranslated regions or UTR, untranslated regions or UTR. And this is the part of the messenger RNA where genetic codes are present. This is the 5' end and this is the 3' end. At the 5' end, there is a cap. Methyl guanosine triphosphate cap is there here. This is the cap. Methyl guanosine triphosphate. Okay. And beyond this cap, there are certain untranslated regions, UTR here. This is called 5' UTR. 5' UTR. This is not translated into protein. Okay. And at the 3' end, after the genetic codes, after the stop codon, the last codon is the stop codon. And after the stop codon, there is a tail. This is the tail. that is polyadenylate sequence. We had already seen that one. Polyadenylate sequence. And after the polyadenylate sequence, again there is a short stretch of UTR, untranslated region. That is a 3' UTR, 3' untranslated region. So these untranslated regions are not included in the protein, or these regions does not this is not translated into a protein, but somehow they influence the translation process. Okay, so that is about mRNA. And from this translated region, this is the translated region where meaningful genetic codons are present. That is to be translated into sequence of amino acid in the protein. Okay. Then the second component in the translation process is transfer RNA. Transfer RNA. It is also called soluble RNA because it is the smallest RNA present in the cell. Transfer RNA or soluble RNA. And this transfer RNA, as you can see in the diagram here, it has 4 lobes. It has 4 lobes. 4 lobes are present. And it appears like, it looks like, the shape of the transfer RNA looks like the leaf of a clover plant. The leaf of a clover plant. Leaf of a clover plant. Clover leaf. Clover leaf, as it is, as the diagram given here. So the model of the transfer RNA proposed is known as cloverleaf model of transfer RNA. Cloverleaf model of transfer RNA. Okay. When we look at the cloverleaf model structure of the transfer RNA, it looks like a double strand structure. It looks like a double stranded structure. Complementary base pairing also you can see. But actually, RNA, transfer RNA is single stranded. RNAs are single stranded. Transfer RNA is also single stranded. But the same strand, but single strand is folded upon itself. The transfer RNA is folded upon itself to form complementary base pairings wherever possible to give a double stranded appearance. Actually, it is single stranded, but folded upon itself to give a double stranded appearance, and complementary base pairings are also there wherever it is possible. Okay. Now, out of the 4 lobes, the 2 lobes are important. The first lobe is an arm. It is called amino acid acceptor arm. Amino acid acceptor arm. And it has a terminal sequence. It has a terminal sequence CCA. A terminal sequence CCA. That arm with the terminal sequence CCA is the amino acid acceptor arm. It is called so because amino acids are bound to this arm. Or a transfer RNA carries amino acid from the cytoplasm to the site of protein synthesis with the help of this amino acid acceptor arm. Okay. And the opposite arm is called the anticodon arm. It is called the anticodon arm. And this anticodon arm, you can see here, the anticodon arm has a 3 base sequence. similar to the triplet codon, a 3-base sequence can be seen. And this is called anticodon. This combination of 3 codons is called anticodon because this codon is complementary to the codon on the messenger RNA. It is complementary to the codon of the messenger RNA. And that by using that anticodon, the transfer RNA reads the codon of the messenger RNA. For example, suppose this is the anticodon for the amino acid AUG. And we should understand that for every amino acid, the transfer RNA are very specific in their function. Transfer RNA are very specific. They are amino acid specific. That means for every amino acid, there is a separate transfer RNA. And moreover, not for every amino acid, but for every codon, separate transfer RNAs are there. So that means there are 61 codons. There are 61 codons. Out of those 64 codons, 61 codons represent some amino acid. So there should be 61 transfer RNA. So for every codon, every genetic code, there is a transfer RNA. Okay, out of 64 codons, 3 are stop codons. They do not represent any amino acid. And 61 codons represent some amino acid. So there should be 61 transfer RNA. Okay. And suppose, so there should be a transfer RNA for every amino acid, of course. So methionine, for example, methionine, the code for methionine is AUG. We know that the code for methionine is AUG. What is the specialty of this codon, AUG? AUG is also the start codon and it represents methionine. Methionine is the initiator amino acid and the code for methionine is AUG. So AUG is the initiator codon. Okay. And this AUG, for the AUG, the transfer RNA which is capable of reading the AUG should have complementary bases at the anticodon arm. So what should be the complementary base here in this transfer RNA? Suppose this is the anticodon of the transfer RNA, then what should be the bases in the anticodon? It should be U, complementary of adenine is not thymine, it is uracil, U, and A, C. Okay, so UAC is the anticodon of transfer RNA which carries the amino acid methionine, because only this anticodon can read the code for methionine, that is AUG. Okay, so during translation, the process of translation, the transfer RNA would accomplish a temporary base pairing with the codons of the messenger RNA. A temporary base pairing. Okay, so for that it should be complementary. So the codon for methionine is AUG and the complementary anticodon is UAC in that transfer. So that means every transfer RNA has an anticodon basis which is complementary to those codons which represent the amino acid that should be carried by the transfer RNA, respective transfer RNAs. Okay, so that is the function of the anticodon arm. which bears a complementary bases complementary to the codons of the messenger RNA, so that during translation it can make a temporary base pairing. Okay. So that's about the transfer RNA. The function of the transfer RNA is to combine with the amino acid, combine with its own amino acids, because every transfer RNA has its own amino acid. So it combine with its own amino acid and carry the amino acid to the site of translation. So that is the function of transfer RNA. Okay. Now the next component in the translation process is ribosome. You know, ribosomes has two subunits. Ribosome has two subunits. So the eukaryotic ribosomes and prokaryotic ribosomes are different in their sizes and slightly their functions. So in eukaryotes, the ribosome is 80S ribosome. The ribosome is 80S ribosome, and it has two subunits. One is 40S subunit, a smaller subunit, 40S subunit, and a larger subunit, 60S subunit. So total it would make 80S subunit. Okay. Then in prokaryotes, the ribosome is 70S ribosome, and the subunits are, the smaller subunit is 30S and the larger subunit is 50S. So a smaller 30S subunit and a larger 50S subunit together, that would constitute a 70S ribosome. Okay. So that is about ribosome. So as you can see in the picture here, there are two subunits, larger subunits and a smaller subunit of the ribosome. And these ribosomes are made up of ribosomal RNAs. The major component of the ribosome is ribosomal RNA. And what is the function of this ribosome? This ribosome provides the site of translation. One function, it provides the site for translation. Or the messenger RNA should be attached somewhere and should be placed somewhere for translation. So ribosomes provides a platform. It provides a platform or a site for translation process. Second thing, it provides certain enzymes and factors. For translation. Okay. And the larger subunit of the ribosome has two sites on it. One is the P site and one is the S site. And these two sites are important in translation. One more site is the exit site is also there. So actually three sites are there. One E site, that is exit site. One is P site and another is S site. So these three sites are important in the process of translation that you would see shortly after. Okay. So these are the major components. mRNA, transfer RNA and ribosomes are the major components of the process of translation. And apart from this, there are factors and enzymes required for translation. One major enzyme required for translation is amino acyl transfer RNA, amino acyl transfer RNA synthetase. This is one enzyme that is required for the combination of amino acid with the transfer RNA. For combining an amino acid with the transfer RNA, this enzyme is required. Amino acyl transfer RNA synthetase. Then second enzyme is in the ribosome. Second enzyme is provided by the ribosome, and that is peptidyl transferase. Peptidyl transferase. This is an enzyme that makes the peptide bond, that establishes a peptide bond between two amino acids. So in the process of translation, amino acids are added one after another to form a long polypeptide chain. So in that process, the amino acids are connected together by means of peptide bond with the help of this peptidyl transferase enzyme. So these are the enzymes required for translation. Okay. Now let us come to the actual process of translation. Process of translation. Let us see how translation takes place. The first step in the process of translation. The first steps in the process of translation. The first step in the process of translation is charging of amino acid. Charging of amino acid, or it is also called amino acylation. Amino acylation of transfer RNA. Amino acylation of transfer RNA. Charging of amino acid. So for the combination or for combining an amino acid with the transfer RNA, at the amino acid acceptor site, acceptor arm of the transfer RNA, ATP is required, energy is required, and this energy is provided by ATP molecule. Energy is provided by it. So amino acid combines with the ATP molecule, energy is provided, and these charged amino acids can combine with the transfer RNA with the help of amino acyl transfer RNA synthetase. That is the enzyme that we have just now seen. So amino acyl transfer RNA synthetase is the enzyme which helps to combine a charged amino acid, an energetically charged amino acid, with the transfer RNA. So that is also called amino acylation of transfer. That means combining of amino acid with the transfer RNA is also called amino acylation of transfer RNA. Amino acylation of transfer RNA. So an amino acid combines with the transfer RNA. As we have seen, transfer RNA are very specific. For every amino acid, there is a specific transfer RNA. Amino acid can combine with only that transfer RNA. Okay, for methionine, a transfer RNA that is called MET transfer RNA. For tryptophan, there is another transfer RNA. For arginine, there is another transfer RNA. So for every amino acid, there is a transfer RNA for that amino acid alone. Okay, so that amino acid can combine with only that transfer RNA. As in the case of genetic code, as we have seen in the case of genetic code, one amino acid is coded by different triplet codons. So in the same way, for one amino acid, for one transfer RNA, there is only one amino acid. For one transfer RNA, there is only one amino acid. Same as in the case of genetic code. One genetic code, one triplet codon codes for only one amino acid. In the same way, one transfer RNA carries only one amino acid specified to it. Okay. So that is the first process. That is the first step in the translation process. Charging of amino acid or amino acylation of transfer RNA. Now the second process, second step in the process of translation is combination of messenger RNA with a ribosome. Combination of messenger RNA with a ribosome or attachment of mRNA with the ribosome. mRNA with the ribosome. Combination of mRNA with the ribosome. Okay. So here, suppose this is the messenger RNA. This is the messenger RNA with the codes on it. The small subunit of the ribosome attaches first with the messenger RNA. This is a small subunit, and the small subunit gets attached with the mRNA. So large subunit would join the team later. Okay. So first, the small subunit of the ribosome gets attached with the mRNA. Okay. Then the third step is actual process of translation begins now. Translation. Actual process of translation, or we can subdivide it. The process is initiation of translation. Initiation of translation. Initiation of translation. Initiation. Actual translation, actual process of translation or the polymerization of amino acid has 3 steps. Three steps. One is initiation. Initiation. So, in the initiation begins when the first transfer RNA carrying the first amino acid. Which should be the first amino acid? The first amino acid is the methionine. So that transfer RNA is called MET transfer RNA. MET transfer RNA. MET transfer RNA. That carries the methionine. That carries the methionine with the anticodon. With anticodon UAC. With anticodon UAC, because the codon is AUG. So complementary to that, the transfer RNA with anticodon UAC combines with the methionine, and transport the amino acid and comes to the site of comes to the site of protein synthesis or translation. So messenger RNA, the smaller subunit of the ribosome has already combined with the messenger RNA, and here it has AUG codon, and the smaller subunit combines with the messenger RNA at the AUG site only. At the AUG site. It recognizes the AUG site because this is the initiator codon. The ribosome can recognize the AUG site, and the smaller subunit of the ribosome gets attached with the mRNA at the initiator codon. Okay. Now a transfer RNA with the methionine, carrying methionine, comes to the site of protein synthesis. And with this anticodon, suppose this is the anticodon arm, this is a transfer RNA, and this is the amino acid. This is the amino acid methionine. So by using this anticodon, it establishes a temporary attachment, a temporary base pairing with the codon AUG and remains there. So first amino acid has come to the site of translation, that is methionine. Now, that is initiation. That is the process of initiation. And after this has happened, after the first transfer RNA has established a temporary base pairing with the first codon of the AUG codon of the messenger RNA, the larger subunit of the ribosome arrives to the site. Larger subunit arrives to the site and combines with the smaller subunit. And I told you, the larger subunit has two sites on it. A P site, this is the E site, X6 site is there, P site and the A site. So the larger subunit attaches with the smaller subunit. The larger subunit gets attached with the smaller subunit in such a way that the transfer RNA fits into the transfer RNA, the first transfer RNA with the methionine fits into the P site. So that is initiation. Now the second step is elongation. The second step is elongation. During elongation, a second transfer RNA comes to the site with a second amino acid. What should be the second amino acid? That depends upon the codon here. Okay, that depends upon the codon here. So by reading the codon, the transfer RNA with the anticodon to that codon, comes with its amino acid, and so this is a site. Okay. And the second transfer RNA comes like this and get attached with the messenger RNA, and it carries an amino acid here, and second amino acid. Now two amino acids are there. One is the P site and other the S site. Okay. Now the enzyme peptidyl transferase establishes a bond here. A peptide bond here. Okay, a peptide bond here. So a dipeptide has formed now. A combination of two amino acids has formed now. Okay. Now the entire ribosome moves forward to the extent of one codon. That is called translocation of ribosome. So the entire ribosome, both the large subunit and the small subunit, together moves forward one step, one code ahead. Okay. What will happen then? Okay. Here Now the site. So it has moved one step ahead. Now this has become the P site and here the S site. Okay. Now this transfer RNA would come to the exit site. This is the exit site. So this transfer RNA would be detached. This transfer RNA would be detached from the protein synthetic site, detached from the messenger RNA, and that would leave the site. Okay. Now a new transfer RNA would come to the S site. It would establish a temporary base pairing with the mRNA, and it would carry, of course, it has an amino acid here. An amino acid here, and the peptidyl transferase would establish a peptide bond between these two amino acids. Now a chain of 3 amino acid. We get a chain of 3 amino acid. Okay. Now again the ribosome moves forward, a step forward. Ribosome moves a step forward, a codon forward, and this transfer RNA would be released from the messenger RNA, and a new mRNA would come to the, a new transfer RNA would come to the S site of the ribosome. So only the first transfer RNA would come to the P site of the ribosome, and the remaining all the future transfer RNAs would be coming to the S site. So this process continues. This process continues, and we get a long chain of polypeptide, a protein, a polypeptide chain would be formed. Okay. So that polypeptide chain would contain amino acid. The sequence of amino acids in that polypeptide chain would be according to the codons of the messenger RNA. Okay. So in that way, a polypeptide chain is produced. Now the third step, the last step in the translation process is termination. Termination. So what is termination? It is the ending of translation process. So translation process ends when the stop codon arrives. Stop codon, there are 3 stop codons. UAA, one stop codon. UAA. When UAA, the stop codon is reached, when the ribosome reaches the stop codon, translation ends. And certain termination factors also help for this. Of course, there are initiation factors to initiate the translation, and there are termination factors to terminate the translation. And these termination factors operate only when the ribosomes reach the stop codon. Okay. And the polypeptide produced will be released into the cytoplasm for further processing. Further processing are required, and for further processing, the polypeptide is released in the cytoplasm. Okay. So this is the process of translation. And the polypeptide thus formed would act as an enzyme to catalyze a biochemical reaction that would result into the phenotypic expression, result into a phenotype. Okay, so this is the content of central dogma. This is how a gene or a DNA segment that can also be called as a gene expresses a phenotypic character. So I hope you understood the translation well. Okay, we shall wind up the class here.